Welcome everybody back from lunch. Hope it was wonderful. This is the AI and Animal Research panel. So we have some wonderful presentations prepared for you. Just want to say thank you for coming back, and I'll do some introductions of everybody so we know who we have the chance to listen to. A quick introduction myself. I'm not particularly important here, but my name is Adam Cook. I'm a 3L at GW Law with Dean Hessler. Our first speaker is going to be Rebecca Critser. This will be on video since she wasn't able to be here for in live. She's an assistant professor at Johns Hopkins Bloomberg School of Public Health and Engineering Department. As part of an interdisciplinary team that brings together lawyers and scientists to advocate for the advancement of new approach methodologies in order to improve public health and reduce the use of animals in science. Our second panelist is Ricardo Tieghi. Tieghi, I'm not going to get that. He's going to have to help me. And he is a scientific program analyst at Dynavet Solutions, supporting the National Institutes of Health. He leverages his position to integrate AI into regulatory policy and harmonize efforts that replace animal testing across all 27 NIH institutes. He's received over 15 prestigious research awards for his work, including the Best Paper of the Year award from the Society of Toxicology's In Vitro and Alternative Methods Specialty section. And our final panelist is Dr. Shagun Krishna. She is the Director of Toxicology at the Physicians Committee for Responsible Medicine. She leads initiatives to implement human-specific test methods that replace animal testing. She's been recognized for several awards, including the Lush Prize for Young Researchers, HESI Cardiac Safety Committee Early Career Seminar Award Series, bit of a mouthful, and Computational Toxicology Specialty Section Yves Allery Diversity Award for Trainees and Young Investigators by the Society of Toxicology. And to begin, as a reminder, since I'm sure you've seen this already, but each speaker will go for 20 minutes, and we'll have 25 minutes of Q&A available here and online, and I hope you enjoy.
Hello. My name is Rebecca Critser, and I am excited to be speaking with you today at this conference on AI, animals, and the law. Let me start out by apologizing for not being with you in person. It was certainly my intention to be there with you this weekend, but apparently parental duties had other plans for me. So I want to send a special thank you to Kathy, Joan, and Jamie, and all the other folks at GW who helped put this conference together, but especially for helping me bring you this content and this material in a format that we weren't originally planning on. I'm really excited to be presenting alongside my fellow co-panelists today, focusing on AI and its applications for animals used in research and testing. And I'm going to start by giving a legal primer. So by way of background, I am a lawyer who works in the academic space. I am based at Johns Hopkins University as part of the Toxicology Policy Program. And I have the good fortune of also teaching a couple animal law courses at the Robert H. McKinney School of Law in Indianapolis and the Lewis and Clark School of Law in Portland, Oregon. I do have to, I'm going to do my intro first. Okay. So real quickly, my team at Johns Hopkins is a toxicology policy program. We are a group of legal and scientific experts who work to advance non-animal models and through various forms of government and advocacy. And you can see us pictured here. Our team is ever growing. Here we are at the DC facility. I do have to give my standard disclaimer that any views expressed in this presentation are my own, and they do not necessarily reflect the views of the university or the Hopkins Health System. So let me start off by making sure we're all on the same page and understand some of the fundamental basics around laboratory animal law. At the outset, it's important to know that there are really two buckets these laws fall into, sort of broad, general, high-level concepts here. One is really focused on laws around using animals in the lab and their welfare. And the other is really focused on the development and the implementation of non-animal models that many see as a replacement for the use of animal models. So to start off, let's talk about some of these laws that really govern the use of animals in research and testing. And as I said, they do focus on welfare. Now, these laws are going to be older than the NAMS laws because we've been using animal models for a longer period of time. The two main federal statutes you're going to want to be aware of are the Animal Welfare Act and the PHS policy. Many of you are probably familiar with the Animal Welfare Act. It is a law that governs the use of animals in many different arenas, not just in a laboratory setting, but it is certainly a major statute, an important statute for knowing when you are doing work with animals in research and testing. It is overseen by the USDA, the United States Department of Agriculture, and it provides species-specific welfare mandates that are sort of a minimum welfare requirement, which you can find in the animal welfare regulations. It also requires that the facilities that use animals in testing have what is known as an IACUC, an Institutional Animal Care and Use Committee, and that some people refer to as an ethics committee. Now, on the other hand, we have the PHS policy, which is not itself a federal statute, but it is authorized and comes into being through the Health Research Extension Act of 1985. It is itself a policy, and it creates certain requirements that are mandated for those facilities that receive federal funding and that work with animals in research and testing. Unlike the Animal Welfare Act, this is overseen by a different federal agency known as OLAW, or the Office of Laboratory Animal Welfare. The PHS policy explicitly requires compliance with the Animal Welfare Act, and it also requires compliance with what is known as the guide, which is where you will find the 3 R's, which we'll talk about here in a minute. Real quickly, I do want to point out the PHS policy is much more focused on sort of self-regulation and self-oversight. Many people would say the same thing about the Animal Welfare Act, but there is at least to some extent a mandate that the federal agency go in and review and inspect these facilities and ensure compliance through that on-site inspection mechanism. So before we move on, we do need to talk about the difference in scope in terms of what animals are covered between these two federal laws. I've already mentioned that the Animal Welfare Act applies broadly to the use of animals in research and testing in the United States, whereas the PHS policy is limited in scope to those who receive federal funding. But there's another really important aspect of this scope definition, which is how they use the meaning of the word animal in these laws. So under the Animal Welfare Act, I've provided you with the very long definition of animal here. You will see it does not necessarily comport with a general or a scientific understanding of the word animal. You can sort of summarize this as being consistent with warm-blooded animals, but really importantly, it does exclude birds, mice, and rats bred for research purposes. And as I'm sure many of you know, the vast majority of animals used in research and testing are in fact birds, rats, and mice bred for research purposes. So there's a large section, a large grouping of animals that are impacted by animal research and testing that are simply not covered by the Animal Welfare Act. Now, under the PHS policy, they refer to animal as any vertebrate animal. So, we have warm-blooded under the Animal Welfare Act. Here we have vertebrate animal. So we're still leaving out some very important non-vertebrate animals. For example, an octopus does not have a vertebrae, but we are bringing in mice, rats, and birds bred for research purpose or otherwise under the PHS policy. As I mentioned, the laboratory animal laws focus on welfare. These are not laws about sort of the ethics of using animals. These are laws about how do we minimize pain? How do we minimize distress? What does adequate food and shelter look like? For some species, what does adequate play or socialization look like? It's really a welfare focus. Okay, so I'm going to talk a little bit about the ethical oversight, the 3 Rs, and the IACUCs that I previously mentioned. Now, an IACUC is, again, that sort of layman's term, General Understanding and Ethics Review Committee. It is mandated by both the Animal Welfare Act as well as the PHS policy. Here we go. So here are the 3 Rs. We have replacement, reduction, and refinement. Replacement is the idea of taking out the animal models and using a different tool to get the data and the information we need. Reduction is about using fewer animals, as few as are necessary. And refinement is about using them in a way that minimizes pain and stress. There are many people who would suggest the 3 Rs are not, in fact, an ethical framework. They were created by Russell and Birch in 1959 and widely adopted across the scientific community by the 1980s. Now, I've mentioned that they are mandated by the Guide for the Care and Use of Laboratory Animals, which is itself mandated by the PHS policy. Interestingly, you will not find the 3 Rs directly mandated by the PHS policy itself, nor the Animal Welfare Act. You'll certainly find the ideas of replacement, reduction, and refinement in the AWA as well as the PHS policy, but you won't find them explicitly being called that in those laws. So on that note, what is it that the Ethics Review Committee is permitted to do under the federal US laws? So they're required to have certain personnel as part of their committee. They're required to do twice-a-year facility inspections, and they're required to review animal use protocols. However, their review of welfare is limited in scope. They can ask things like, have you thought about ways to minimize pain? Have you considered whether or not we have the proper facility to house these animals? And researchers are required to address whether there are available alternatives to an animal model through this committee review. However, the committee members are not permitted to ask questions about scientific validity. They are not allowed to inquire or to question whether the design of the experiment itself is sound, or perhaps if we've done this experiment or other facilities have done it 20, 30, 100 times, do we need to do it 101st time? Essentially, you can't do a pros and cons analysis. What the committee is really there to do is to sort of ensure that the researcher is meeting the regulatory requirements and providing these minimal welfare standards. So in talking about moving away from animal models, we're talking about NAMs and we're talking about funding. NAMs is a shorthand. It's not necessarily defined consistently across agencies, but generally it stands for New Approach Methodologies. And we just call it NAMs for short. So I say here that NAMs is not necessarily defined the exact same way across agencies. This is a really important consideration. We use animals in research and testing in a lot of different ways. And those different applications have different legal and regulatory oversight. So basic research is primarily governed by the NIH. Chemical safety analysis is generally covered by the Environmental Protection Agency, the EPA. And then, of course, we have cosmetic safety as well as drug safety. And in the case of drugs, you also have efficacy, which are governed by the Food and Drug Administration. Each of these are, their authorities stem from different statutes, which have different implementing regulations. And so when we look to investing in and developing an appropriate regulatory framework for the approval of NAMs, we have to think about all of these different legal structures. I do want to call attention to this coordinating committee known as ICVAM, the Interagency Coordinating Committee on Validation of Alternative Methods. They came into being in part to help the different agencies that oversee animal use think about, develop, and integrate NAMs into their work. It is important to note, however, that ICVAM does not have any authority to mandate actions of the other agencies. They really are designed to coordinate, to disseminate information, to house information, and to support the other agencies. They cannot mandate certain rules or requirements. Also, the OECD, which stands for the Convention on the Organization for Economic Cooperation and Development. This is the international body organization that operates sort of similarly. The OECD works widely on sort of regulatory testing and produces guidelines that are generally accepted throughout the world at a very sort of high standard. And so getting the OECD on board to adopt a regulatory approval framework is also going to be an important part of international harmonization. So in addition to thinking about the regulatory framework for validation approval, we also have to think about the database, sort of where do we access the information? So TSAR is one database. There are multiples that houses new approach methodologies that either are currently going through an approval process or have been approved by one or more governmental agencies for use. So the TSAR is maintained by the European Commission. And you can see here it has contributions from a variety of countries, including the United States. I think I've included here an example of what one entry looks like. So this is one NAM that has been entered into this database, and you can follow along its process from submission, currently, or at the time that I snapped this image, was in validation phase, and then it's peer review, recommendation, and regulatory acceptance standards. A NAM can't get to that final step of regulatory acceptance standard until a governmental agency, at least one, has given it regulatory approval. So changing gears a little bit here to some of the legal developments in this space. The FDA Modernization Act was an effort that succeeded. This bill has passed, become law, to change some of the language in the Food, Drug, and Cosmetic Act that talked about preclinical testing. So preclinical testing is all the testing that happens before someone can move their drug testing phase into a human, to clinical testing. So clinical means human, preclinical means everything before human. I cannot explain to you, maybe somebody can explain to me why during this revision, this FDA Modernization Act 2.0, they changed the language from preclinical to non-clinical test. Generally speaking, preclinical is the word that is used, but currently non-clinical is the word that is in the statute as it stands today. So there was a requirement before that said that to get drug approval, to move to clinical testing, you had to finish sort of preclinical testing. And it had, I think, in parentheses, including, animal data or something to that effect, which to a lot of people didn't necessarily require animal testing. It just said if you had animal testing, that was included in a preclinical test data batch. But some people interpreted it the other way, interpreted it to mean that actually animal testing was required. Now, this revision makes it clear that non-clinical test data can include data from in vitro models, which are sort of generally speaking in the lab, in a test tube, in a petri dish outside of a living body. So in vivo is in the body, in vitro is outside. In silico models, these are going to be your computational models. This is where AI would fall into primarily, although not necessarily exclusively, or in chemical models. And this is where instead of working with tissues or cell in a dish, you're working with just sort of a chemical to evaluate. And then, of course, you still have the animal model as an option. It says, or a non-human in vivo. So this revision did not remove animal models as a means of doing preclinical or non-clinical testing, but it explicitly makes clear that other models are available. We've also had the FDA roadmap. And actually, maybe I will just, for the purposes, there we go. Which was sort of the FDA coming out and saying officially, we understand the importance of new approach methodologies of NAMs. We understand the limitations of animal models, and we want to begin moving that forward. Here's how we're going to do that. And I think my colleagues might talk a little bit about more details about what it is they're going to focus on. I wanted to highlight for you just these two pieces, which is step 3 and 4 of their plan, is to establish a validation and a qualification pathway, and then to develop regulatory guidance and standards. And that's really where we are in this phase of moving from an animal model to new approach methodologies, including AI models, is that we need a framework. We need a framework that allows for this transition to happen, that considers the myriad of ways in which we use animals for testing and data gathering, and in a way that makes sure that these new tools, these NAMs, are in fact giving us reliable data. I just briefly want to talk about the fact that there is a lot going on in the legal space around NAMs right now. I think in 2025, there's something like 16 federal bills that have been introduced, which is just around animal testing. It's a very large number for animal testing in 1 year. So there are some efforts to end funding for animal research, some bills that address ending funding for animal research generally and sort of widely, some that look to do it within the government itself, so saying the government shouldn't be spending money on this. Others that look to say, we shouldn't be doing this outside of the US, shouldn't be funding animal testing outside of the US. We also have bills like the SPARE Act, which is a very large sweeping bill that looks to end animal testing, and I think something like 3 to 5 years. And I want to talk briefly about some state laws as well. So a lot happening at the federal level, but equally interesting, there's a lot happening at the state level. So I think it's something like 35 bills have been introduced at the state level just this year. But rather than talk about a bill, I want to talk briefly about this Maryland law. So in 2023, Maryland passed and enacted the Maryland Human Relevant Research Fund. This is a fund, a mandated fund that takes a tier-based approach to assessing a fee against those facilities that use animals in testing based on the number of animals they use in their work. The money that is received from this fund, from this fee, is then used to support the development and implementation of NAMs. The purpose of this law, as stated, is to promote state-funded research intended to develop human-relevant alternatives to using non-human animals. And that, of course, would include AI models. And so this is a really interesting sort of test case to see if it can, this sort of fee structure can be part of this larger transition from using animals in research to moving to new approach methodologies, including AI models. And with that, I will conclude. I have my information listed here. If you have any questions for me, please don't hesitate to reach out. Again, I apologize that I can't be with you there in person. And I want to, again, thank the folks at GW and my co-panelists for all that they've done to make this happen.
Wonderful. And now we'll hear from Ricardo.
Wonderful. Thank you all for having me today. Is there... So hello everyone, my name is Ricardo Tieghi. I am a contractor supporting the National Institute of Health Division of Program Coordination, Planning, and Strategic Initiatives. And today I'm going to be sharing some of the recent events that have happened launched by the NIH and the FDA, supporting the advancement of new approach methods. Just a brief disclaimer, my opinions are my own and do not represent any of the agencies like the NIH, Dynanet, or the FDA. So I know it's an after lunch presentation, so I will do my best to keep you engaged. But it was in 2021 when I first became involved with animal research. And I joined the heart disease lab, and day in and day out, I would go to their mouse facility, and I was cutting toes, cutting tails, and euthanizing dozens of animals, and specifically mice, on a daily basis. And you can imagine how that sat with me. The experiments, they didn't feel useful, they didn't feel like we were extracting the maximum amount of information. So that's when I joined the NIH, and I left that lab to begin working with AI and computational methods. And that's when I began to understand the problems of animal testing. And that's really when the question flipped in my mind from, why would we want to replace animal testing? When perhaps maybe the question should be, why are we still doing animal testing? As you may know, some of the problems of animal testing is that the US is one of the largest users of research animals, probably in the world. And especially in the drug development industry, approximately 50% of that research is not reproducible. When you factor in the economics of the situation, it means the US is wasting about 28 billion every year and a countless number of animals in the process. And when you consider that the animal-based data has significant implications and failures to predict the human response, this means that we're slowing down the drug development process and putting all of us at risk. And so, with those considerations in mind, over the past several years, there's been a significant push to advance the use of new approach methods, or NAMs, as Rebecca described. And recent surveys have found that there is a strong public bipartisan support behind the advancement of these methods. And just as an example, the regular use of NAMs, such as organ-on-a-chip technology, in this case it was by Emulate, for liver-on-a-chip technology, could save the industry up to $3 billion annually. So today, I'll demonstrate some case studies of how the NIH and the FDA are truly working to phase out the use of animals and phase in the use of these new approach methods. And so what are NAMs? Just quickly building off of Rebecca's presentation, these can entail a wide series of technologies, and really they're all-encompassing non-animal techniques, such as computational modeling systems, AI. They can include organ-on-chip technologies, or simple cell-based assays which are not using animals. And here's a sample image of what these look like. And currently, our preclinical or non-clinical testing pipeline relies heavily on animals. A pharmaceutical drug company will use NAMs extensively during the drug discovery phase. However, due to these outdated animal testing guidelines, they have to test their drugs on animals, which they will submit to regulatory agencies and then move on to human clinical trials. However, our current testing paradigm has one large fallacy. 90% of the drugs that actually go on to human trials will never be approved for human use, and they will not receive approval by the FDA. So with those considerations in mind, the FDA launched in April of this year their roadmap to reduce the use of animal testing in the preclinical testing pipeline. And so some of the considerations of those include, and the FDA and the NIH are partners in this effort, and in their document they discuss extensively how they will walk together from the process of identifying which endpoints do we actually need to replace with these new approach methods? Where are the animals tests failing us? And essentially, they will walk through together on how they will develop the validation and qualification pipelines to introduce NAMs into the regulatory policy. But also, how will we drive a culture change within the NIH and the FDA so that reviewers become familiar with the new approach methods themselves and can understand their underlying data? And so the FDA in this roadmap, they actually pledged to revise their internal policy guidelines for industry, but also the international ICH guidelines that support decisions that pharmaceutical sponsors have to submit for animal testing. The FDA also has pledged in its roadmap to incentivize industry to submit new approach methods data and promote the success stories when NAMs are used successfully in place of animal studies. And so if you consider all of these factors, the outcome of, or the new revised preclinical pipeline will become something like this, where the drug discovery companies will use NAMs in the discovery space, but also to validate their outcomes. And here we can use the NAMs not as a one-to-one replacement of animal tests, but we can begin thinking of them as a tiered strategy or weight of evidence approaches, where you may have one in silico and one in vitro method replacing a single animal test. And then we'll use animal testing where NAMs are unavailable or perhaps not fit for a specific endpoint. And so the NIH's vision of NAMs is really this interoperable, integrated environment where they're not used as side projects to animal testing, but instead they are foundational to how we understand disease, biology, mechanism. And it's really about creating the future of biomedical research, which is grounded on human relevant technologies which are scalable and can grow effectively. And so in 2022, the advisory committee to the NIH director put together this working group report, which was adopted in 2024. And their key takeaways were that to truly realize the promise of NAMs at the federal government, we need socially responsible technologies, we need comprehensive training of reviewers, and we need coordinated infrastructure that will truly drive this institutional change. And this is essentially the key takeaways here is we need to integrate all of this simultaneously to realize the promise of NAMs. And so how is the NIH doing that? Here is some of the current existing efforts by the NIH which support this vision of NAMs as suggested in 2024. This includes the advisory committee itself, but also the NIH is part of ICVEM, which was mentioned by Rebecca, which is this large interagency effort across the US federal government. The NIH has also launched landmark initiatives such as the complementary program, which I will discuss further, and cross-center advancements in tissue engineering and organ modeling across its 27 institutes. Very interesting, perhaps, the NIH has also recently, in one of its workshops, announced how the NIH will adapt its funding language. So the NIH is now prioritizing funding of new approach methods. Future funding opportunities will no longer exclusively mention animal testing and will consider NAMs in their appropriations. And essentially, here is just one example of a very large-scale initiative which was very transformative in the NAM space when the NIH launched the Complementary Initiative. They awarded $1 million in a prize pool to 20 different teams using computational and experimental techniques together to develop the future of NAMs. Perhaps most interesting, they also awarded a series of funding programs for new technology development centers and NAMs data hubs that will house the NAMs in the future once they are ready for regulatory application. Another recent AI initiative at the NIH is the real-world data platform. And here the NIH is aggregating information from large-scale data sources such as claims and billing, electronic health information, as well as pharmacy data, and even sensors and wearables to truly drive current longitudinal studies, inform drug development, and even help with real-time health monitoring. Another one of the flagship tools at the NIH, which I highly recommend you investigate, is called the Integrated Chemical Environment. It's a part of the National Toxicology Program. And this is a tool very useful for method developers in the NAM space, where they can identify reference chemicals lists from highly curated data sets. You can put your experimental results in the context of human exposures and run all of our machine learning models for toxicity. And it's all meant to be used by all of us in this room, and they're all extremely user-friendly. Another up-and-coming initiative at the NIH is the Collection for Alternative Methods for Regulatory Application, and the government loves its acronyms, so this one is called CAMERA. And in this case, it's going to be a centralized repository which will serve two main purposes. The first one is for regulatory agency. It will be the centralized hub of all validated methods that come from complementary and all other initiatives. And it will contain all of the training data, their validation data, their repeatability across different labs, and it will enable researchers and the regulators who are reviewing these NAMs for approval to truly understand if those NAMs are suitable for their context of use. However, it will also be extremely useful for the method developers and the pharmaceutical industry themselves, as it will contain all of the language from the regulators about which NAMs are acceptable and for which endpoints. And we intend to make this a graphical user interface which is easily accessible. So just quickly building off on Rebecca's presentation, ICVAM is the Interagency Committee on the Validation of Alternative Methods. It's a mouthful, but there are 17 different federal agencies which are a part of ICVAM, and they've really been driving the change behind NAMs over the past 20 years since they began to become federally mandated in the early 2000s. And perhaps one of the most important documents they've put out in 2024 is the Validation and Qualification documents, which introduce the concept of context of use. And this is for method developers that want to create technologies for regulatory application. You need to specifically consider what type of chemicals are you using, what agency will it be submitted for, which center, in which context. And there's very other considerations, and they've masterfully created this graphic which represents sort of the 6 key points from the document just on the cover page in case you don't feel like reading it. And another recent exciting update from the NIH is that just the end of last month, they announced the Standardized Organized Modeling Center. And this is really a truly first of its kind initiative across the US federal government, where they are creating across all the NIH institutes an attempt to resolve one of the biggest challenges of organized modeling by creating this Standardized Organized Modeling Center, as there's a very critical issue with standardization across labs. And so this is an effort aimed at resolving that situation, and it's going to be extremely useful for pharma, the extramural research, and biotechnology in the future. And so now I'll pivot slightly to the FDA side of the equation, and earlier this year, the FDA put out their initial recommendations for how to develop AI for regulatory applications, and it's a short document I recommend you check out. I will summarize it later. And building on that, they recently released an extremely exciting resource at the Center for Drug Evaluation and Research, FDA CDER, on 50 drug development contexts where NEMs are actually either allowed or they're under active consideration. And this is a table you can access at the QR code. And for example, for safety pharmacology, they introduce in which cases would NEMs be approved and even encouraged for submission instead of animal data. And this is a short list of short-term priorities from the FDA, obtained from the FDA Director, Clinical Assistant to the Director, Tracy Beth Hogue, during one of her recent presentations about some of the priorities where we will be replacing animal testing in the near-term future. I won't dive into too much detail, but now I will announce some of the more exciting, perhaps, updates on AI at the FDA. Towards the middle of this year, they launched one of their first large-scale AI initiatives at the agency, and they called it ELSA. Like most tools nowadays, they have unique names. And it was meant to truly solve a lot of the challenges at the NIH or at the FDA and accelerate how reviewers can compare drug labels, how they can navigate and summarize data, among other challenges. However, Jeremy Walsh, during one of the chief AI officer at the FDA, said there are still challenges that must be addressed. For example, the FDA has historical data on animal testing since the late 30s to 40s. And he estimated there's about 300 to 400 million paper records of the FDA. So how do we go about addressing this challenge? The good thing is these problems are solvable, and they're solvable today with AI. So this is just an effort that, for example, we developed at the NIH about how you can automate data extraction going from hundreds of unstandardized, unstructured files, PDFs, scientific publications, whatever content they may be, and you can use large language models and machine learning to get them into an automated, standardized tabular format and get high traceability of your results by annotating the original PDFs. This was recently published in Wired's Computational Molecular Science. And here I ultimately wrap up with just a review of all sort of the key regulatory considerations method developers should have when considering NAMs for regulatory application. This includes everything from beginning and critically analyzing the agency's needs. Does the agency actually have a fit for your NAM in a regulatory context? Can you collect a reference list of chemicals to test your methods? And this includes both AI and other general NAMs. Can you demonstrate that your assay, AI model or not, is it reproducible across labs inside your own laboratory? And can you demonstrate that its performance matches or exceeds that of the current animal testing it intends to replace. And then you obviously can submit it to the FDA, and it will promote your success story once it's accepted. And so to wrap up my talk, I just wanted to quickly overview some of the opportunities supporting NAMs and funding opportunities that may be of interest. Recently, the FDA launched its Common Fund Replication Prize, which aims to address the challenge of replicability I mentioned earlier in my presentation. So we partnered with NASA's Tournament Lab, and you have until December 19th to submit your ideas. It's $850,000 in funding, and multiple teams will be awarded prizes. So I highly recommend you take a look at this challenge prize. And secondly, to address the time cost it takes to develop and validate NAMs, currently it sits at about 5 to 10 years from development to validation of NAMs and integration into regulatory pipelines. The NIH launched the NAMs Reduction to Practice Challenge, and this will enable us to, for NAMs that are near to production, they will enable us to learn from their challenges and how to develop the frameworks that will be used for all future NAMs once they are ready for implementation. And the deadline for that is March 1st. And so just in conclusion, and to wrap up my talk, our current preclinical testing pipeline uses animals, but recent efforts at the NIH and FDA to prioritize NAM drive the culture shift that will motivate and integrate NAMs fully into the preclinical testing pipeline, will eventually realign animal testing and drug development with a human biology-based paradigm. With that, I'd like to acknowledge Dynanet for the support and my mentor, Dr. Nicole Kleinstrauer, as well as our FDA colleagues, Tracy Beth and Martin Macri, as well as PCRM for the support. Thank you for your time, and I'm happy to take any questions.
It's wonderful. And now we'll hear from Dr. Krishna.
Okay, thank you everyone for having me here, and thank you very much for Kathy. Thank you very much to Kathy for inviting us and organizing such a great panel. And it's my honor and privilege to be here and talk about a topic that sits right at the intersection of innovation, ethics, and science. So my talk is mostly about how we can utilize artificial intelligence-based approaches to drive and to build a smarter, faster, and innovative framework that's not only gonna be human relevant, but also ethical to animals. So every year, millions and millions of animals are still used to test the safety of the products that we rely on. And these products, they range from cosmetics to drug molecules to all the environmental chemical that we have been exposed to. And we have been exposed to tons and tons of environmental chemical every day. And by default, from decades, the default form or the default way of testing those molecules is through the animals. But imagine a world where we can predict the toxicity of any chemical or any drug molecule without harming a single chemical, where we can make decisions based on the information that we gathered based on the human data. Because at the end, we are not 120 pounds of animal, we are not 120 pounds of rat or 120 pound of dogs. So that's exactly where artificial, and again, the opinions are just mine, and that's exactly where I feel that utilizing these human relevant new approach methodologies are gonna take us. And again, I'm not going to reiterate the definition of new approach methodologies. I'm pretty sure that each one of us in this room now know that what are new approach methodologies. So, okay, but again, I will do because this is my favorite topic. So, new approach methodologies are anything that doesn't use animal, and in my opinion, which is smarter, faster, and more ethical. So, and these are few of the tests that we use every day on animals. And I just want to remind you that my talk is mostly focused on how we utilized animal in toxicity evaluation of different chemicals and how we can utilize these AI-based approaches to move away from using animals in the chemical testing. And here in this list, you can see that what are the drawbacks of utilizing animal, and what I feel is most important is, in addition to the ethical concerns, that they are very poor predictor of any human health outcome. And and I'm pretty sure that you have learned from Ricardo's talk and it's so great to see this, that now FDA regulators are speaking the same language. They acknowledge during different recent events that animal models are poor predictor of human outcomes. Okay, and in this slide, I'm going to just show you that what are the new approach methodologies look like. As I mentioned, that those are the future of toxicology screening. They consist of different components. That's how I characterize them. So there are how we can move away from animal testing. What are the alternatives that are available to us? One is in vitro, like how Ricardo mentioned. These are the miniature cell-based system that actually mimic the human organ. And of course, they have been derived from the human cells, so they are human relevant. And then comes the in silico methods, which are basically computational-based methods. And that's where the AI, artificial intelligence, and machine learning-based models comes to play the role. And then we have the omics technology, means like we actually sequence our whole genome, and not just the gene pool of an entire human species, but also the protein pool and all the metabolites that are inside our body. And that field is called metabolomics. So the omics field is evolving very rapidly, and they are actually going to help us to be at a point where we can utilize the human-based methods for the real decision making by the regulators. And then we have in chemical methods that uses different chemical reactivity assays. So for examples, from decades, researchers have used rabbit or mice to test the skin sensitization of all the cosmetics. But now they have been replaced by methods that have been derived by human skin models. And one of these methods is called DEPRA. And what is the impact of using these new approach methodologies in the regulatory decision making or during even in our research? As I mentioned, that these are faster, they are very time effective, they are cost effective, and of course, they are based on human relevant decision, and they are sustainable. Okay, and this slide is actually gonna take you to the different approaches that we use. These are the different AI-based methods that we used to derive those workflows. Like Ricardo mentioned that we, as researchers, we build different workflows that we present to regulators and then they accept it. So these are the different AI approaches that nowadays have been used frequently for the toxicological applications. And one is machine learning. Machine learning is basically where we learn from the data. We identify the patterns from the data and then we make the decisions. Think of machine learning as a recommendation system of your Netflix account. If you're watching a lot, I'm like, that's a super simplified example, but I'm pretty sure that everyone could relate to that. If you're gonna watch a lot of horror movies, you will get recommendations for the same horror movie. So we train our model and then it gives us predictions. So we mostly use these kind of methods for the first, or for maybe like for first or second tier of screening, where we screen chemical for a particular toxicity. Then comes the deep learning, as the name suggests, deep learning. So it's a complicated form of machine learning based method where we utilize different layer of bless you where we utilize different layers of information to make the decision. And there are different deep learning based approaches, neural networks, and then convolution neural network, and there are multi-layer neural networks. I'm not going to go deeper into that, but I think in my subsequent slides I'll share you a couple of resources where you can actually read and learn more about this. And then comes the natural language processing, like how the Ricardo mentioned, that how FDA is using the ELSA tool. So basically they are going to extract the chemical, like in the toxicological reference, we extract the chemical effect relationship from different historical data. Like he mentioned that we have a ton of historic data available to us, and it's really very difficult to mine or to synthesize the useful information out of that data using manual methods. So there comes the role of natural language processing. And then we have computer vision, which basically detects the subtle cellular changes in imaging data. So think of it like you have MRI images. So a model that has been trained by different MRI images that is actually able to detect the changes in those images and can predict the outcome. And then we have the knowledge-based system, which actually combines the expert rules with the data-driven models. Okay, and then, so across what goes inside these AI models. So the AI models are not just a black box. We provide them useful data, and the quality of those models depends upon the data that we provide them. And here are a few examples of the data system that we use for the toxicity prediction of any chemical. So there are high throughput repositories. US federal government has an initiative of ToxCast and Tox21 that has tested thousands and thousands of chemicals across 3,000 assays, and these databases actually provide a very useful and reliable resource for generating AI-based model to predict a certain kind of toxicity. And I'm going to present you a case study. I hope that I'll have time, that how we can utilize these data and how we can utilize these different approaches to actually make decisions for a certain subset of chemicals. And then again, this one is like how I mentioned that we can use omics data, and then there are a lot of chemical data. The chemicals come with their inherent property, like they have certain molecular weight. Each and every chemical have a unique descriptors through which we can represent them. So we utilize chemical data for building these AI models, and then patient data to derive the human-based decision. People can use the different clinical records that contain valuable patient information, and that way they can actually derive the decisions that are personalized, that are basically that are unique to human species. Okay, now let's take more closely that how these AI methods are making their room in the toxicology space. So as I mentioned that AI is transforming toxicology, not just how we analyze those huge data sets that are coming out to every day, but also how we make decisions. So this is kind of like, this is different ways through which we can make decisions by using big data, by using all the data that is going into the AI-based model. So it starts with knowledge integration, where AI rapidly connects insights across thousands and thousands of resources, or maybe study documents, or even the regulatory documents that have been submitted to FDA or EPA. And then we are using it for risk assessment. We rapidly merge evidences from different data streams, and which actually help us to refine the testing strategies that are more human relevant, and that way they are kind of like reducing, not kind of, they are actually reducing the use of animals. And not only this, we can also utilize AI to run some virtual clinical trials. Like, we can create digital twins, like we can simulate the whole human body and see how a chemical or a drug behaves inside the human body. And shared data structure, like AI curated databases are there, which can be shared across different, not only between different companies or different, but across the globe. And I see AI not just as a scientific tool, but also as an ethical opportunity, that how we can utilize AI in toxicology. So in this slide, I'm going to go over few key pillars that actually guide AI use in toxicology. So first of all, and again, this is my favorite and this is my priority, to minimize animal use in testing. So AI directly supports our ethical imperatives by reducing the animal testing while also improving the human health protection. And I feel that each study or each AI model that we create for a certain type of toxicity actually a step forward toward a more human and more human relevant science. And then bias mitigation. AI models can learn what we feed them. So if our training data is biased, then of course the model is going to predict the outcome according to that. For example, if I'm building an AI model based on 10,000 chemical for a certain toxicity, say cardiotoxicity, out of those 10,000 chemical, only a handful of them are showing cardiotoxicity, that we call positive, and then others are not showing cardiotoxicity, that means they are negative. So our data is imbalanced, and eventually our model will be biased toward negative chemicals. So that is one thing that we have to be very cautious when we are building AI models. And then transparency and explainability. So AI can't be black box, as I mentioned. So we have to be very transparent about the data that we are using for these AI model. Like in Rebecca and Ricardo's talk, that the data sharing principle has to be established across different regulatory agencies or different different countries, so that if we want to create a harmonized ecosystem where we can use one AI model, that is going to be like, that way that's going to be more acceptable if we are very transparent and explainable about what is going inside the. Oh God,
Yeah.
so sorry.
No, you're good.
Yes.
Oh, there we go.
There you go. Okay. And finally, the human oversight, AI, and I was, before joining to PCRM, I was working with Dr. Nicole Kleinstroyer, and she came up with this, with a new definition of AI as augmented intelligence. And I totally agree with her that AI should always augment human expertise. And so the toxicologist and the human expertise must be in loop when we are using the AI models. And any technology's true impact comes when we use it for real world case scenarios, or where the regulators trust and adopt these methods. So how we can build trust among different regulatory agencies so they start using these AI based methods. And I know that that's already happening. We are actually witnessing an era which is very revolutionary, and we have been seeing the change that we wanted to see from decades at least. So the first step is early and continuous engagement. If you want to build, and that comes with any NAM based method, not just the AI. Like even if we are working on an organ on chip method, or even we are working on an in silico method or a computational model or any AI model, I feel that if we involve regulators at the early stage, that's going to be super helpful, because this builds mutual understanding and avoids any surprises later. And we know that what is their need, what has to be done so that industry and other companies start using that methods. So for that purpose, I think it's really great to involve or collaborate with regulators to be on the same page. And then second is defining the clear context of use. We have to clearly, when we are using or when we are building AI models for the toxicity testing, we have to clearly articulate that whether an AI model is meant for screening purposes or prioritization or the risk assessment. So this clarity helps to align the validation with the real world decisions. And third is the mechanistic understanding. So the regulators like FDA or EPA, they need to see that the model actually reflects the biological responses. It's not just the data driven. It actually, the data that is building, or the data on which we are building the AI model, has to be biologically or mechanistically driven. There are, I'm not sure how much we know about the in vitro models in this room, but so if we are making an AI model based on some high throughput screening data that is used for the toxicity screening, we have to have a biological response behind that. And then international uncertainty qualification, which I feel that is really important, because all these high throughput screening data where we test thousands and thousands of chemical in very short time, they always come with uncertainty. So we have to counter that uncertainty when we are building a model. And I'll show you a case study that how we can incorporate that uncertainty when we are building an AI model. And then, of course, international harmonization, like how Rebecca mentioned, that there is OECD, which is Organization of Economic Cooperation and Development. OECD works a lot on non-animal based methods. We at PCRM work very closely with OECD to not only build methods that uses non-animal that uses non-animal data, but we also pushed them to make a guideline so that it can be accepted by each and every country. So if OECD issues a guideline, a test guideline that, okay, we are not going to use animal for the skin sensitization, that means that all the member countries of OECD has to accept that test guideline. And finally, the collaborative governance. We have to encourage partnership among regulators, NGO, academia, and industry people so that we can guide the responsible AI integration. And ICWAM is doing a great job in that. So ICWAM annually hosts its public forum where they share their work throughout the year, and where we as a nonprofit or non-government organization or any research organization can submit their comments, that what changes you want to see, or based on their work, if you have to provide any feedback, you can provide the feedback on that. So as I mentioned, that we are really in an era where we are seeing the changes are actually happening. Okay, and these are few of the challenges that we see for the NAM-based methods. And okay, I'm gonna quickly move because I really wanted to show you a case study that how we can utilize these AI-based methods. And these are the opportunities that how we can advance the validation of AI-based NAM. So we have to use a standardized validation framework. We need to benchmark the data set. And again, the transparency has to be there. And again, the NAMs are not just a single method, like how Ricardo mentioned, they are basically a battery of different methods. So hybrid modeling approaches, collaboration, and data sharing. And this is how the future of AI-based toxicology looks like. And I would really encourage you to scan this QR code because it will lead you to a research review article that has been written by Dr. Nicole Kleinsteuer and Dr. Thomas Hartung, and that's going to really take you to a deep dive of how you can utilize AI for the toxicity testing. And then I'm going to share a case study that how we utilized it in real world scenario. So as I mentioned that we have been exposed to tons of chemical every day, and these chemicals affect our cardiovascular system. Few of the risk factors are very clearly established, like smoking, air pollution, and lifestyle, but then the chemical exposure, that's where I'm interested in. So we work with HESI, which is Health and Environmental Science Institute, to come up that how a cardiovascular fails to perform its life-sustaining functions. They come up with the idea that, okay, fine, we can categorize it into 6 different types. And these are mentioned here: change in vasoactivity, action potential, like how your heart rhythm, and things like that. I'm sorry, Okay, so I basically wanted to show you the workflow, that this is how we move from tests that are done in laboratory to make decisions that are human relevant, how we incorporate the different data that I explained before, and how we incorporate different methods of these AI to actually make real world decisions. So we start with all the chemical assay, the high throughput screening in vitro data, and we combine them with the cardiovascular failure modes. Then we identify that which is the most sensitive bioactivity. And as I mentioned that the in vitro data always comes with uncertainty. There are ways to incorporate uncertainty in that, and that has been developed by EPA. So there is a great ToxBoot package in our program that you can utilize to incorporate uncertainty in the data. And then I utilize the ICE dashboard, which Ricardo mentioned, which has been developed by the NIC Atom, to build the dose that we need to be exposed from the amount that we are seeing in the in vitro assays. So imagine that you tested a chemical in a dish, but then how you can make human relevant from the outcome of the assay that is in a dish. So we extrapolate it, and you get the dose that is required by humans to be exposed to see those adverse effects. And then we compare, And then I even compared it with the traditional animal studies, and we found that the dose that we are deriving using these NAM-based approaches are way more protective than the traditional animal studies. And then we have human exposure data through different databases. I compared the doses that we obtained from the NAM-based methods to the human exposure, and we found the margin of safety that we need to create. And by using these methods, we identify few chemicals which are actually still in market, but we feel that they are potentially harmful. So this is not a workflow that has been currently accepted by regulator, but I'm just giving you a flavor that this is how we utilize different data and different approaches to make regulatory decisions. And I'm gonna quickly go through my conclusion again. I feel that AI and NAMs are driving us to a faster, smarter, and more ethical future. And I want to thank my mentor, Dr. Nicole Kleinstrer. When I was at NIC Atom, she used to supervise me, and the case study that I performed here was done when I was at NIC Atom. And thank you all for being here. I know this is the last talk of my session.
Thank you, thank you. I at least found that absolutely fascinating. So we'll take some time to ask some questions. I'll have the chat open here for people online if you'd like to ask questions. And I had some of my own, but we're running late on time, so let's just jump here.
Thank you for the presentation. Thank you for the presentation. So one thing I heard from a few researchers or advocates or NAMs is that NAMs are often measured or validated against how well they match the animal models. So as a way of validating whether they are suitable for being used in drug discovery, they measure how well they match the performance of animal models, and that seems totally wrong for me, and wonder if you agree that is wrong, and if that represents another evidence that they're reluctant to give up animal models.
I have some input on that. Actually, it's a great question. And there has been a paradigm shift over the years about how can we demonstrate that we're better than the animal test, exactly if we're comparing to the animal assay itself. And so for specific endpoints, and we've used skin sensitization in the past, that's the potential of chemicals to induce allergies, for example. How can we demonstrate that we're better than the animal assay? Well, in this case, we have large databases of human testing. And so when the human data is available for a particular endpoint, we will always give prevalence to how can we compare the AI or the NAM model against human testing of that chemical with like a reference list, for example. But when that data is not available, if you don't have human data, then we do compare it to the predictive performance of animal testing. And in that case, we just want the method to demonstrate that it's as predictable or better than the animal model. I don't know if you have any thoughts.
Yeah, I would just like to add, I think I'm audible enough. Okay, can you hear me now? Yeah. Okay, I just want to add that yes, we use the traditional animal data, but not for the validation, to compare that how NAM-based predictions or how AI-based predictions are better than the traditional animal studies. We do validate them. I mean, like as a researcher, I always prefer to validate them against the human data. But yes, I always compare them to animal data just to showcase that how the NAM-based predictions are better than the animal data. And that's what I showed you in my case study.
But what about the regulators?
Sorry?
Do the regulators require the NAMs to be measured against animal models or human data?
As of now, I think yes, but that's where we are seeing the paradigm shift. And it's so great to see that they are very open to validate them against the human data. And that's where, and I think none of us mentioned that, and that's where FDA's iStand programs come in. So they have this, the iStand program, and I'm so sorry, I cannot think the full form on top of my head now, but that actually helps to validate the in vitro data without using the animal data.
Great. Other questions back here?
This is a little bit along the same lines. One of the challenges we recognize in veterinary medicine is cross-species physiology and comparative physiology. And you mentioned that humans are not 120 pounds of mouse. We often will say a dog is not a small human, a cow is not a large dog, and it's very true. And it got me thinking about, even in testing, how important it is to have models that represent the species that is the end point to be represented. So having human relevant data for humans, having bovine relevant data for drugs for cattle, et cetera. And I'm fascinated by seeing how we can develop more of those models so that we are actually meeting the needs of the different species. I wondered if you could speak to that a little.
Okay, so I think because of lack of my time, I couldn't cover it, but we have now physiological based pharmacokinetic models. Those are actually species specific and they integrate all the NAM based approaches. They are built on the NAM based data. So there you can provide species specificity, and we can actually make decisions based on a specific species. So but yes, again, that has not been accepted by any regulators, but through several programs, I know FDA is encouraging the use of those models. And we are working with OECD also to accept those approaches for the regulatory adoption.
Other questions? Anyone back here?
Hello. You spoke briefly, or multiple times, both of you, about in vitro and organoids. I'm curious if there's any legal frameworks around brain organoids specifically, because I've seen you can now commercially, for $35,000 or so, order 800,000 neurons that's already connected to a computer, or even use brain organoids through an API out of Sweden. This seems kind of unconscionable to me, and I'm wondering what your thoughts are on it.
Yes, it's an excellent point. I will note that, for example, the NIH is investing in the Standardized Organoid Modeling Center. Although their priorities are not brain organoids, they're focusing on heart, liver, and other systems. And the reason for that is because there is a very developed understanding of brain organoids and a whole institute devoted to that at the Johns Hopkins University. And so I can think of a presentation I saw from Dr. Lina Smirnova, who's one of their leaders in brain organoids and their interface of using AI with brain organoids. And they've connected organoids to computer games to assess drug development. So that's just one example I can think of. I'm not exactly, I can't fully answer your question. I'm not intricately familiar with the brain organoid technology, but I do think we're going to see a lot of progress in that space. And I don't think there are any regulatory frameworks set in place, but I do think the outputs in the coming months and years from such as the Standardized Organoid Modeling Center will greatly standardize how organoids are developed and also ensure that they're being applied to the drug discovery pipeline. Hopefully that answers your question.
So is your question motivated by the worry that brain organoids might be sentient and might be able to suffer?
Yes. So I've seen a few examples of research like you're describing, running an LLM on a brain organoid and then giving it an embodiment in the shape of a virtual butterfly or a robot in some cases out of various institutions. And so, yes, my concern would be that it's a form of conscience and that we are enslaving them to fill a purpose.
Yes, that's an excellent question. It's not something I had previously considered, but I unfortunately don't work very closely with brain organoids, so I don't know if I have a great position on your statement, but it's an excellent consideration. Yeah, thank you for the question.
Is there a question up here? Or across first, sorry.
Here and then.
Okay, yes.
And I'll happily pass the microphone to whoever. Just on that, there's the ISS CR, the International Society for Stem Cell Research, and they put out guidelines for stem cells as well as extensions of them that are voluntary, but that kind of are the biomedical lab standards, and they have talked a lot about chimeras as well as organoids. So I would look up ISSCR as sort of the framework where a lot of that research, and Hastings' report is also very good for this stuff. Just want to say thank you to everyone who's your pioneers and you're advancing this, and just for the whole, everyone involved, you know, if it's okay, I can just take a quick second. This is a long time coming, right? Very recently, Peter Singer and I wrote an op-ed called a Paradigm Shift to Animal Testing, and in that he wrote, you know, 50 years ago in animal liberation, I advocated for, or he advocated for, the use of alternatives to animals. And there was maybe what some people call the most effective animal activist of the 20th century, Henry Spira, who in the 1980s led campaigns with full-page New York Times articles that had rabbits and tests on them. And those campaigns against Revlon and against the American Museum of Natural History ultimately led to the founding of the Center for Alternative to Animal Testing at Johns Hopkins, CAT. Revlon gave $750,000, Chanel, Bristol Myers. So people have been working on this a long time, and the FDA Modernization Act was bipartisan. Rand Paul and Cory Booker were the two advocates. This is bipartisan, this is popular, and it's starting to win. So yesterday there was a question of where AI for animals is. This is it. This is happening right now. The NIH, FDA, you're leading it, and there's a real opportunity to celebrate it and to make it you know, institutional. So just want to say thank you. I just wanted to bring one other thing and ask a question. We spoke a lot about academia, government. You know, ultimately we want drugs that work, that are cures for people, and all people, human and non-human animals. And what about the industry side? You know, there's Roche has started an Institute of Human Biology. Vividyne has raised $40 million. Axiom in toxology is ready. So how is there a connection between what you're all doing and getting this to actually into industry? So Ricardo or Dr. Krishna or whomever.
Yes. So interestingly, the industry is welcoming these kind of decisions. I know that it's really hard for them, and maybe because of the cultural issues, or maybe because of their old school mentality, that they don't, because they have been doing something from so long and they just don't want to change it. But again, since we are hearing it from the regulators, and that's in my personal opinion, I think that's the best thing that's happening. We are hearing it from the senior leadership of NIH. We are hearing it from the senior leadership of FDA. So if the regulators won't require the animal testing, eventually the industry people will not do that. So that's why I mentioned in my talk that this is a wonderful time for people like us who are pushing for this from years, at least for me, not for decades. So I think that if it is coming from regulators, industry is going to welcome. I'm pretty sure there are few people who won't like the change, but they have to come. They have to come on board at some point of time.
Thank you, Sankapa, for the beautiful comments and the excellent question. I think ultimately the decision in industry comes from a financial standpoint, right? They will be motivated to incorporate NAMs if there are financial incentives or if eventually it translates into cost savings for them. And I think we're developing tools currently at the NIH and the FDA to translate the costs of these NAMs. So if we start incorporating NAMs, how much are they expected to save in an IND submission, for example? And we're developing calculators internally to provide to industry that will enable them to save time, save money. How much do we project these cost savings to be? And I think this will be a massive tool for the industry, because if you have the financial incentive and the regulatory updates simultaneously, this will be a huge driver behind the adoption of NAMs by industry.
Yeah, thank you.
Hi, thank you for your talks. They were really interesting and insightful. I'm wondering what your thoughts are around biases that have been kind of discussed to exist, either within kind of editing boards for journals or also within funding groups. So there's some literature that I've read about this fairly recently, which talks about researchers who have been trying to publish papers, and they have mainly or just non-animal methods. There's no kind of animal testing that's been done, and they've been asked either to validate their results with animal models, or their papers haven't been published in kind of more well-known journals, but also with funding boards that fewer research has been kind of funded if it solely consists of non-animal methods. So just wondering what your thoughts are around that, and whether you know of anything that's going on currently to kind of combat that bias.
Yes, I'll take this one. Thank you very much for bringing this out. So we at PCRM, we have a program that is called the Coalition of Eliminate and Address Animal Methods Biased. That's CoLab program. It has been led by my wonderful coworker, Dr. Catherine Crabb, which actually addresses the biasness of including animal methods in funding opportunities or even in publishing. So we are actively gathering surveys from not only in the United States, but in different countries. We recently have a survey had a survey from India, and I think that paper is published. So we are actively engaging with researchers to speak up against this. And I think initiative like that is actually going to mitigate the bias that is currently a part of scientific ecosystem.
I have also a point on that. I've heard similar stories about papers being rejected and requests for animal data, but considering that the NIH, for example, is one of the largest funders of biomedical research in the world, if their future funding opportunities are all centered on, not all centered on, but they're including NAMs and this incorporation, I think that this change is not purely administrative, but with time, there will be this paradigm shift where most or practically the majority of studies will begin to have consideration for NAMs, for alternative technologies. And I think, unfortunately, here, it's apart from initiatives like Shagun and PCRMs, it will come with time that will begin to address these biases.
Oh, yes. And I would like to mention one more thing. On 7th July 2025, there was a conference, there was a workshop for FDA and NIH. And during that workshop, Dr. Nicole Kleinsler, she announced that NIH is not funding projects exclusive on animal models, and now they have to include language of NAM. So I think that initiative like that is really encouraging, and they are going to support researchers not to use animals, and they're going to also encourage the reviewers who are actually reviewing the publications or the funding agencies not to send people or not to push people to use animal methods.
Awesome. And we'll go ahead and call it a wrap there. Join me in thanking our panelists, please.
Thank you very much for having us.